Thank you very much for being here, Scott, and for sharing this with us. We really appreciate it.
Okay, thank you, Eduardo. And oh yeah, good. Okay, so we are recording. Can everybody see this?
It's perfect.
Okay, so yeah, what I'm going to do is kind of give a little bit of a background about who I am and why am I here at BioSummit and doing the things that I've been doing. And it's kind of weird that Google Slides, the up and down arrow doesn't work, so I'm going to have to futz with the mouse trackpad. So yeah, a little bit about myself. Okay, so I'm one of the old farts. I've been around for a while, and I actually started off, I lived in 3 countries and 5 cities before I turned 14. The last, prior to Canada, I basically grew up in Australia. So I actually started undergraduate at RMIT University in Communications and Electronic Engineering. And in fact, you can see the date there. It's, it dates me. But this image is actually from it was a wind cheater, a t-shirt competition for designing the logo for the department. And it turned out my submission won, so they loved it. And so a little bit of art design history from my past. So I did a couple of years of communication and electronic engineering, then switched into biotechnology. And I completed actually my year was the first year for the biotechnology program. And I'm not certain if it's still running at RMIT now. So from Australia, I came to Canada, to Vancouver, and I actually worked in research institutes, both in Australia and here, before starting up graduate studies. So in Vancouver, I was at the Biomedical Research Center, and then I started up a PhD program in genetics at UBC.
You're a little quiet to my ears. Can you maybe speak up very slightly?
Okay, how is that? Is that better?
Yeah, or increase your volume just a little. Thank you.
I don't know if I can control. How is that? Is that better? Testing 1, 2, 3. Okay, so I did my graduate studies and I worked in academia. I did a postdoc in Toronto at the Ontario Cancer Institute and the Amgen Research Institute. And this photo on the left here is an image actually of the Celera war room, essentially. So Celera was the company that sequenced the human genome. And this is me, I don't know if you can see my cursor, this is me sitting in the front of it. And these people were flown to Celera to participate in their Human Genome Jamboree. So when I was up in my postdoc, I was doing a lot of bioinformatics research, identifying genes in the human genome raw data that was coming off of this Celera platform. So that was a lot of fun. And on the bottom right here, there are the 2 publications that came out, the first draft publications of the human genome. So Science on the left was Celera, and Nature on the right was the public effort. And really, for all of my life, I've had this fascination for this spinning thing here, this thing called DNA. And it's been a real passion. And I think, you know, I keep saying, biology is awesome, and I'm in awe of evolution. Okay, so I'm going to go to the next slide. In 2015, a group of us actually started up the Open Science Network Nonprofit Society. There's originally a group called DIY Bio Vancouver that started in 2010, and I started talking with Eric Zepka, and I don't think he's at BioSummit this year, but I think he's been at all 3 in the past. My understanding is he's still in Bangladesh, probably under lockdown. But he and I instigated the whole thing to get the ball rolling. And we got an amazing group of people to help with this. So these are just some of the images. I won't go into them in the detail. I will say, so that the top right, this is at the BC Genome Sequencing Center. So we had a tour of that. But in the center, I've had my whole genome sequenced. It's up for anybody to download. You guys will have to figure out which one on the personalgenomes.ca site. So this is the Canadian version. George Church, Harvard geneticist, started up the US, or the first personal genomes program. I think they have over 350 people genomes. Yeah, you can try to figure out who I am. Bottom right corner, this is one of my passions that my colleague Wes Wong does, and it's the STEM Mentoring Cafe. So it's one of our community outreach programs. It's where we get STEM professionals to sit with middle school students and try to encourage more grade 8 students into STEM fields. People have called it speed dating for STEM. So the kids come in, they talk to the mentors for about 15 minutes. They go from table to table to table, doing things like that. So people in previous sessions here, we've talked about sustainability. Well, when you do things like this, when you organize that, you end up getting people recognizing that. And so on the left, here's a check for 5,000 Canadian dollars that we received from the Ted Rogers Community Foundation. And these two young women in lab coats flanking the check, holding up the check. I mentored them in a program for the science fair. So they won the top mark for the Vancouver District Science Fair and a silver medal for the regional. Okay, you guys are here for, okay, next step is when you start up things like this, you never know where it's going to take you. And I was in the lab in mid-2016, and the founder of Maker Labs, where our lab is located, it's a for-profit makerspace. He asked me if I'd be interested, if I knew anybody that would be interested in doing the synthetic biology course. And I said, I'd love to learn more about the course. What type of person were you thinking about? And he said, I was thinking of you. And so I ended up taking the How to Grow Almost Anything synthetic biology course in 2016, and I acted as an instructor. I think it's still operating, but it's gone internal to MIT. But that's how I met David and George. And so 2017, Global Community Biosummit 1.0. And the rest is kind of history for Biosummit. Everybody's here, everybody knows about it, and it's just been amazing. And in Biosummit number 1, you know, I came thinking, how can I contribute? And of course, I do photography, so I picked up my camera and just started taking photos. And so I've created a body of work, and I've been back at all 3 Biosummits. And a lot of those photos are actually on the biosummit.live website. So if you go into the top right corner, there's fun stuff. Go in there. They're on Google Drive. You can actually click on the images, share, you can comment, you can do what you want with them. And so in 2018, FreeGene's I learned about FreeGenes, and that was another transformational step. And then in 2019, I learned about Open Bioeconomy Lab, and I'm going to go in a little bit more into that. So FreeGenes, BioBricks, many of us have heard a lot about FreeGenes over these last 3 days. In 2017, Drew Endy with his BioBricks Foundation, he received funding to be able to synthesize off-patent or IP-free synthetic genetic parts. And it turns out, so Isaac Larkin here on the right, at the bottom right, he's the one that came up with the name FreeGenes, and Keane has been a big driver of FreeGenes. He left FreeGenes at the end of last month, and he's doing some amazing stuff with Bacillus subtilis and genetic parts there. So he's both these guys are people to watch. Okay, what's next? Open source DNA. So what I'm talking about is the Open Yeast collection, and it's a collection of DNA parts. And I've been passionate about open source software. I'm not a particularly good programmer, but I've been interested in it almost from the beginning. And I understand the history of open source software and the power that it has had in transforming the world that we have. So, of course, I'm completely interested in something that allows the transfer of DNA and other biological materials. It's not just for DNA, for anybody to use, unencumbered by any laws, rules, regulations. Countries have their own laws that govern access to this. So I know, for example, New Zealand, they have restrictions. Europe has restrictions in many places around the world. So if you can get the free genes, they're free for you to use, but there are also constraints in terms of where you live as to what you can do. So it's free in terms of the DNA, but not necessarily free in terms of other things. But Linda Call here, who was with the BioBricks Foundation, she's the one that drafted the OpenMTA and had input from a great many people. It's a very powerful document. In the Jogel conversation, I showed this image. This was earlier in the pandemic. Jenny Malloy, Chiara in the center. I think people here who were here last year probably attended her workshop on their bioreactor. It's really amazing. Fernan up here, he's just amazing, incredibly knowledgeable. I don't know if he's in here or in another session, but he's really pushed the next iteration of, it's not just the genes, but it's the backbone components in terms of allowing us to freely use the genes that are in the Open Enzyme collection. Isaac, of course, here, and Isaac number 2, Isaac Nunez, who works with Fernan. And this is Mark Fleshman from the BioBricks Foundation. And of course, everybody knows Drew here. Okay, so Open Enzyme collection. So I've mentioned it. It's been mentioned many, many times. This thing is amazing. And yesterday in our talk, I was trying to think of the word, the phrase, context is everything. And it's a phrase that I frequently say. We're restricted by what we can do because of the context that we're in. And Open Enzyme collection is one of those things that can expand our context. So if this is something that's interesting to you, then I'd highly recommend getting it or collaborating with labs that have it. And as Jenny mentioned earlier today and yesterday, this collection is a collection of reporters and enzymes. And this number 31, actually it's up to 84 now. I must have an old version of the slide. And so it's the gene part, oh, sorry, that's over here. Yeah, okay, my mistake, 60 reporters. So this whole collection here of all these parts, so you can think of these as parts, and I'll be talking a little bit about the Golden Gate assembly strategy. So each of these colored blocks is a part, and it's a part that you pull out of your freezer and you say, I want this promoter, I want this tag, I want this promoter, this cleavage site, this gene or CDS, and this terminator. You assemble them in a pot, you mix in your special enzyme, which I'll talk about, and then you build this part. Fernan and Isaac and Chiara and Isaac Nunes, so Isaac Larkin and Isaac Nunes were really fundamental in building the FreeGene strategy for this. I had some peripheral contributions to that, but I'm really grateful for what they've done and what they've put out to the community. Okay, so the Open Yeast collection, I'm going to first give you the 10,000 foot view. It is also a collection of parts, like the Open Enzyme collection. I'm calling it OC1 because I'm hoping there's an OYC2. So maybe people in this audience might want to build their parts and contribute it and spread it around the world. So one of the things about open source software is that people built their software and they shared it. People used it, they remixed it, they reformed it. And now we have the internet as we have now and so much other software. Where will open source DNA go? The FreeGene strategy uses what's called the Golden Gate MolClo strategy for assembly, and I'm going to give more details about that later. Okay, so the Open Yeast collection is an open source. It was inspired by this paper by John Dubner's group at Stanford University. And it is also a MolClo strategy for assembling parts to be able to build metabolic pathways. Mine borrows heavily from it, but it's also different. We're using the FreeGene's assembly standard, and so some of the, it's kind of interesting, every academic comes up with new definitions in terms of how they're going to do the MolClo because it's such a flexible thing. This system is on AdGene, so if you can afford the $300 US plus whatever the shipping is, you can get those DNAs soon or right away. Okay, so now I'm going to step into metabolic pathways. So the Open Yeast collection, primary usefulness is for building metabolic pathways. So if you think of living things as factories, it's there to make biomolecules. And what does that? It's really enzymes. So enzymes are proteins that come from genes that catalyze a reaction. So it takes a substrate, makes a chemical modification, and converts it to a product. So if there are any software people here that are familiar with Unix, it's kind of like a Unix command line tool. A command line tool does one thing and one thing well. Then you pipe it to another command line tool and it just goes from one to the other. So here on the right, I've got a cartoon of a metabolic pathway. So this yellow ball is the first enzyme. It takes the first substrate and converts it to a product. That product is the substrate of the second enzyme, and so on, until you get the final product. And there's a wide range of things. I think I saw that Kevin Chen is here. Kevin Chen, founder of BricoBio. Everybody knows him, Global Community Bio Summit. He has a company where he's using yeast to produce THC. This is Canada, it's legal, and it's not just THC, but also other cannabinoids. So this is a good example.
Kevin is here, Scott, I think he's put his name down.
Okay. So he's doing amazing work there in the for-profit arena, and it's amazing. But the whole concept is that using cannabis as an example, the cannabis plant has its own biochemical pathway that has a whole bunch of enzymes that produce cannabinoids, and then the final one is the THC. And you could stop it by modifying the enzymes, and you can do a lot of different things like that. And yeah, let me go to the next slide. Okay, so pathways can be short. So on the left here, for people interested in possibly making bioplastics, you might want to look at these two enzymes. All of these things come from out in the wild, so either from microbes or from plants or from other species. Genome sequencing projects that have been happening all around the world for microbes, for everything, has really built up this massive database, public database. There are also private databases that we don't have access to. But this pathway on the left is just two enzymes that will build a precursor for a bioplastic. How easy can that get? On the right, if you wanted to make vanilla, the flavor, that's a much more complex pathway. And of course, the vanillin plant does that, and this is how that plant evolved. What did I say about evolution is awesome? Yeah. Okay, so metabolic pathways are many, and this is kind of like a simplified map, and there are a number of resources where you can go where you can explore it. But one of the things about the pathways is, okay, so I talked about substrate going to product. These are biomolecules that are small molecules within our body. There are so many of them. We've got the amino acids that go to make the proteins. We've got the nucleotides that go to make the DNA. We consume sugar. We make lipids. We make all sorts of things. And plants, they have their sets of things, like the enzymes that are involved in taking light and converting that to energy, sugar and CO2 within the plant. So it's a massive area. I don't even know a fraction of it, but I know that we can explore certain aspects of it. And what I want to see as the Open Yeast collection evolves is to see some of the common pathways, enzymes for those being built and then set out for free, and then some of the terminal enzymes, which might be more proprietary or might be more useful. You might want to just keep it for yourself, or if you're interested in developing a company around it. So I see technologies like the Open Enzyme Collection, the Open Yeast Collection as being enabling innovation. As I said, context is everything. If you don't have this, you can't do this. And in the community biology arena, where money is tight, free is awesome. Free genes is awesome. Okay. So why metabolic engineer? These are some of the examples. There are people that are making flavors, and this is actually an important part of the Open Yeast collection. This is just one branch of all of those pathways. So it's called the terpenoid pathway, and there are a lot of different variations on it. So you just pick your enzymes to go from one place to another, and you can produce fragrances, like here in the top left corner. Down in the bottom right corner, you can build pathways that produce drugs. So the bottom right is Taxol. So Taxol is a drug that was discovered in yew trees and used to treat breast cancer. And it's one of the frontline treatments for estrogen sensitive breast cancers. Very important drug. It is synthesized, as I said, in the yew tree. Originally, they were cutting down all these yew trees to extract the Taxol. Then somebody in the chemical, a chemist, had figured out how to synthesize it using petrochemicals. So it was manufactured like that. But now people have figured out how to build that pathway to produce Taxol. very important drug. And just above it, artemisinin, it's an anti-malarial drug, another one that's being manufactured through biotechnology. And there are other things. There's so many things that are possible, and this is just one pathway in the many. So there's a lot to explore, and you can get really overwhelmed quite easily, but you just have to come back to where you are at now and move forward from that point. You know, take a deep breath, be patient, and you can do cool things. And I'm going to get back a little bit later to this part of the pathway because it's an intrinsic part of the Open Yeast collection. Whoops, went too far. Building metabolic pathways, we assemble the DNA for all of these different enzymes, and then we put it into yeast to do what we want. Or some people put it into other species, like there are pathways in bacteria, pathways in algae, all sorts of things. And actually some of the components of the Open Yeast collection are very useful for people working on those other species. The Open Yeast collection will be the first non-prokaryote or non-bacterial kit collection in the FreeGene's portfolio, let's call it that. But with here, it's more than just putting the DNA in. There's a fair bit of work in terms of trying to block off competing pathways and things like that. But those are some of the stumbling blocks that we all will be facing as we move forward with this. And hopefully we can build a community that's going to help to solve some of these. And yes, okay, so now on to the Open Yeast collection. So as I said, it uses the FreeGene's Golden Gate strategy. I'll describe on the right here what the parts are. And it's actually been extended. So there's another 2 parts, which are the bacterial backbone. But starting off in this middle section, so we have the promoter here, the gene, and the terminator. And all genes have these. So you need a promoter, which turns on the production of the RNA, which then gets translated into a protein. So that's the RNA is transcribed from the gene. So it's up to you to define what the gene is. Terminators are there to help isolate things, particularly if you're going to be concatenating things into a pathway on a plasmid or something. Okay, so that's the core. All MolClo strategies have that. OpenEnzyme, it makes up this as well as these. What's really new with the Open Yeast collection in terms of an open source system are these assembly connectors, and they are just short bits of random sequence that has these the ability to build the meta plasmid that has the entire pathway. So with the FreeGene strategy, with the MolClo strategy, we talk about levels of parts. So all of these parts are called level 0. So they are, here I'll admit, Heidi, all of these parts are level 0, and you basically just pull out from the freezer what DNA level 0 part you want to assemble, and that first assembly creates a level 1 part. On the left here is kind of like a color-coded view of the parts, but this digs down into the MolClo strategy where we talk about a prefix and a suffix. You don't have to worry about the details of this, but the important thing is that the suffix is the prefix of the next item. So for this assembly connector, we have a suffix that's GGAG, and that part would assemble next to the GGAG, and it's this 4 base pair overhang, which I'll explain in a bit, that defines how these parts assemble in your one-pot assembly. Okay, let's go here. Whoops.
Scott, quick question.
Oh, yes.
Did you want questions that hold questions until the end?
Yeah, I think maybe at the
ahead.
end. I think that would be, and I can move back and forth through the slides because I know this is going to have a lot of questions for people. Okay, so there's a whole series on the Golden Gate cloning up in YouTube. I've got the link here. David's going to paste it into the chat session for you to explore. But don't start watching the videos on the side. Wait until after my talk. But I'm just going to show you the first video, which talks about these special restriction enzymes. So restriction enzymes, and there are a great many of them in the OpenEnzyme collection, both regular type 2 and type 2S. So it's the type 2S that are the special ones. So this, I'm just going to, oops, what happened? It's only 3 minutes. Can you hear?
You need to refigure the audio back through.
Pardon?
Switch your audio over, probably.
Okay, so how do I do that? Is it first time?
I think you switch it back from your headphones to the computer, perhaps.
Oh, okay. So if I pull my headphones out, that should do it.
Welcome to the Safety Laboratory.
Do you hear that?
No, yeah.
The first video
Not very clear.
is about the Golden Gate
Can you
cloning
hear
DNA
that?
assembly method. In this video, we'll learn about type 2S restriction enzymes
I
and
can't
how
see
they
the
differ
chat,
from
so if
classical
somebody can
type 2
call out.
restriction
We do, not very clearly, but we can hear that.
Restriction enzymes cut DNA by making two
Better. Thank you.
to the phosphate backbone.
Let me just start that over
This
again.
is the first
That's
video
good stuff.
about the Golden Gate cloning DNA assembly method. In this video, we'll learn about type 2S restriction enzymes and how they differ from classical type 2 restriction enzymes. Restriction enzymes cut DNA by making two incisions, one in each sugar phosphate backbone. They are classified into types according to their structure and whether they cut their DNA substrate at their recognition site, or if the recognition and cleavage sites are separate from one another. Classical type 2 restriction enzymes are commonly used in molecular cloning. Each enzyme recognizes a specific sequence, usually palindromic, shown in orange. They cut their substrate within their recognition sites, leaving either sticky, that is 5' or 3' overhangs, or blunt ends. When a desired fragment of DNA, shown in blue, is released from a plasmid using a pair of classical type 2 restriction enzymes, part of the recognition sequence, shown in orange, will be present at the ends of the released fragment. This means it can only be ligated to other pieces of DNA that have compatible ends, usually those that have been cut with the same enzyme. When this happens, restriction enzyme recognition sequences will be present between the two fragments. These are known as assembly scars. Golden Gate cloning uses type 2S restriction enzymes, which cleave outside of their recognition sequence. For example, the recognition sequence for BPI1 is GAA-GAC, shown in orange, but the cut is made in the 6 base pairs downstream of the recognition sequence, leaving 4 base pair 5' overhangs. There is no sequence requirement for the identity of bases in the cleavage site. In this example, a fragment of DNA, shown in blue, is flanked by 2 BPI1 recognition sequences, GAA, GAC, in opposing directions, shown in orange. When this fragment of DNA is released from a plasmid using these enzymes, no part of the recognition sequence will be present in the released fragment. It can be ligated to other fragments of DNA that have compatible ends, and when the 2 pieces of DNA are joined together using a DNA ligase, no part of the restriction enzyme recognition sequences will be present. This is the end of the first video. In the next video, we'll talk about...
Okay, so you can go on to watch those videos to learn more about it. And it really goes into it in detail. So where those overhangs were 4 N's, N is the universal code for all 4 bases. So we get to define what those ends are. And so FreeGene has developed this strategy, and I've worked with Keeney and Fernan, Chiara, Isaac. There's this amazing collection of standards. And the whole idea of this MolClo strategy is to really make standardized parts. So all promoters have the same start and end, the prefix and suffix, right? So you go to your fridge, you pull it out, and you know it's just going to work. It's just going to link to your gene. So you can build up a database, an actual physical database of these parts, and you just go and get them. And what I hope that this community can do is contribute to building up that database, not just for the Open Enzyme, the Open Yeast collection, but also for the Open Enzyme collection, because there's so much power. Let's mix and share. It's awesome. Okay, so let's see. So transcription unit parts. So remember, we have a promoter, we've got the gene, also known as a CDS, and then we've got the terminator. So in the Open Yeast collection, I've introduced a whole bunch of promoters. So we have, these are all yeast promoters. So people can pull any one of these out to assemble with their gene of interest. On the right, I've got the terminators. There's a bunch of them. And, you know, again, this system is extensible. So if you come up with a terminator that seems to work better in a certain context, or you think it's just so much better than everything else, contribute that. In the middle, that's the interesting bit. So with Dugner's yeast toolkit, he doesn't have a whole lot of components to be able to build metabolic pathways. His is everything else but this bit. You know, he does have fluorescent proteins. But what I wanted to do when I was designing this, since it's an open source thing, it's going to be distributed, I wanted tools that people can use for learning. You know, once they get it, they can start to work with learning how to do a MolClo assembly to build these things. And then there are other things. So at the top, these are actually, or these ones here from AD1 to AUR1, these are actually, they're genes that are involved in yeast metabolism. But there are yeast genes or yeast strains where these genes have been deleted. So those strains are called oxytrophic strains. So they're missing one of these enzymes. And if you grow them on a media where most of these are for amino acid biosynthesis. So for example, this one, MET17, is needed to produce the amino acid methionine. So yeah, these are important when you want to put genes back in to have selection. You know, it's a bit like the ampicillin resistant marker in a bacterial plasmid when you're working with E. coli. These two genes in the middle are recombinase. I've been interested in recombinases in the past. They both recognize different 34 base pair repeats that you can cause flipping or recombination. Kind of cool. And they're just parts. I may not use them in the context of yeast, but somebody else may. The next two, now these are kind of interesting. So a DNA activation domain and a DNA binding domain. So some people here may have heard of the yeast-2 hybrid experiments. So yeast-2 hybrids are ways to be able to do protein-protein interaction. And the way that works is that you have your protein of interest. So let's just say you've been studying a protein and you want to know what it binds to. Or you have two proteins and you want to see if they bind together. That might be the simplest to explain. So you can make a fusion of one protein to the activation domain and the other protein to the DNA binding domain.
Scott, sorry for interrupting. Just to let you know, we have 3 minutes until the end of the
really? Oh my
session.
God.
Is that okay? Sorry.
I'm going to jump that. Yeah, sorry. I've
No problem at all. Just to let you know.
Yeah. Okay. So I'm going to go. The next bit are the genes that would be interesting here. These ones are for scent. So they're the terpenoid pathway. So this allows you to build the pathway and use your nose, not a mass spec or some other expensive equipment, to be able to produce it. Sorry, time went by. Okay. The level 2 assembly. You can look at the YouTube video to learn about that. So open yeast collection. It hasn't yet been synthesized. I wanted to make sure that the open enzyme collection, the open reporters, and the backbones are there for the community to use. I didn't want to compete with that. And so open yeast collection, all the parts are with FreeGenes, and we're just starting pretty soon. Hannah tells me they'll be synthesized. This is the link for it, but if you just go to stanford.freegenes.org, or I think David, I think I gave you the link, if you can pop it into chat. Okay, so I'm gonna escape out of that, and sorry, I completely lost track of time babbling on, and I'm gonna open it up to, let me stop sharing, to questions.
Thank you
Natalia had a
very
question,
much.
I believe. Sorry, Eduardo.
Nope, go on, Alison, sorry.
Natalia, can you unmute and ask your question, or would you like me to read it?
Hello, sorry. Thank you, Scott. I just want to know if there's all the metabolic genes are already there of the yeast, or there's like a collection of selected genes. This is the first question. And the second one, if we have to use like a specific strain from each assembly, or you can use like a generic strain from everyone.
Yeah, so this is the first iteration of the open yeast collection, and a lot of it really depends on the community at large. So if you have a favorite pathway that you want to build, there's nothing stopping you from using the FreeGenes MolClo standards to design PCR primers to amplify up a gene that you're interested in studying, or the genes within a pathway. So there's a lot of flexibility. Remix, match, share, that's the whole idea. So what I have in the toolkit is basically a basic pathway that allows people to use smell to determine whether their learning process of building the pathway works.
I've actually got a really basic question if we have time.
Well, I guess we can go for this one more question. What do you guys think? And then go on to the break, for the break.
Oh, is it a break, or do we have something coming up?
In theory, we should have a 15-minute break before the next session.
Okay, okay. So I don't mind hanging on if people want to continue on, and of course, if the moderators are willing to do that. And I apologize for babbling on.
Okay, so I've got just a basic question. We're working with Cerevisiae, yeah? Or are we branching out to some of the other yeasties?
Yeah, so this system was designed for Saccharomyces cerevisiae, but those assembly connectors are agnostic. So if you, there's a fellow in Egypt who is interested in this collection because he's working with Yarrow histolytica. And the assembly connectors, they should work in a wide range of species, right? Because they're just short bits of random sequence that have the BBSA1 site for the level 2 assembly. So it really is agnostic. You just have to build the specific parts that you want. But yeah, and then, you know, some of the yeast enzymes will work in other fungi.
Yeah, I guess I would just say a caution from someone who worked a lot with non-model organisms.
Yeah.
It depends on how related your organism is to the model organism in question. It's not always predictable. So I've got a couple of other questions.
Sure.
One from Patrick Hassenfeld, and then there's one from Patricia as well.
Sure.
Patrick, do you want to unmute and ask, or do you want me to read? Can't hear you, Patrick.
My question was, how is FreeGenes and open enzymes connected?
Thank you.
FreeGenes and open enzymes. So FreeGenes is a component of the BioBricks Foundation. If you go to biobricks.org, you can learn more about it. So they received funding in 2017 to start synthesizing these genes, and they put it out to the community. What would you like synthesized? The open bioeconomy is overseen by Jenny Malloy, who's been at many of the sessions here, and she's amazing. She's built this amazing community to be able to connect people, to help people with the open enzyme collection. So her people, as well as collaborators, and I've had a small part in it, have curated a whole bunch of enzymes to be able to have synthesized through FreeGenes. And they are actively using them. So Jenny is a Shuttleworth Foundation recipient. Shuttleworth is the co-creator of the Ubuntu Linux distribution, and he wants to support building the economy in Africa with his money. And so Jenny is bringing the open bioeconomy to Africa. And of course, this plays out everywhere around the world. And she has collaborators right around the world. And you know, people are recognizing the amazing. I'm actually emotional because what she's doing is actually quite powerful for the betterment of humankind. Sorry, next question.
Next question, Patricia, if you can please unmute. You've had your hand raised for a while.
Hi. So I know you mentioned that you can use you can use the metabolic pathways with the the thing to test if two proteins can interact and bind to each other. Could you also use it to see if you can prevent them from binding with each other?
Oh, absolutely. The other thing that you could try is also, if you have the two proteins binding, you might find a molecule that could inhibit that binding. So it's called the yeast 2 hybrid. And if you Google that, there's a lot of information out there. There are commercial kits to do this. They're well characterized. I want to create this open kit that people can use and share. And, you know, if you discover something that you think will go and, you know, allow you to earn a living building a company, all the power to you, you know, or anybody here. You know, this is true open source, unfettered DNA. So yes, the yeast 2 hybrid is a very powerful. You can even, you know, the concept is the bait and the target. So you're looking for protein binding between the bait and the target. Well, you can create what's called the library of genes from a species. So you might have a bait you want to fish out a part from the library. So it's a very powerful tool. And Patricia, you're awesome.
Thanks.
Thank you, Patricia. And as Eduardo mentioned, we have time for maybe one question, so get in fast, folks. Now's your chance. Yeah, Carolyn, I think we answered your question in the chat.
I have another one, if that's okay.
Okay, just a quick one, please.
Scott, very quickly, I'm a bio-artist. I don't have any of these biotech skills. I have some, but I really wonder, where could I learn this? So I'm incredibly intrigued and really want to know how to, use promoters and assemble and stuff like that.
That's actually an awesome question.
Where do I have access to learn this?
Okay, well, one, that video on the MolClo strategy, that gives you the basic overview of the understanding of how to assemble things. And this is a game changer. When I started, it was all that old fart kind of stuff. But that would be a good place for starting. And then I think for understanding a general how to work with DNA, I couldn't recommend, and I think Patricia will be behind me on this, amino.bio. Justin and Julie, who've been at BioSummit in the past, they are amazing. They've created these educational tools in their book, which Patricia is a co-editor on, is called, Patricia, do you want to comment on it?
Ah, yes, it's called Zero to Genetic, I cannot speak today. It's called Zero to Genetic Engineering Hero, and it's actually really good. I'm not just saying that because I helped edit it. It's actually like really good, and it bridges the gap
I can
between.
confirm that.
Please pop a link to the chat and the notes.
Yeah, amino.bio is the website.
I can drop a link to both the website and then the book separately.
Please put it in the chat. And Scott, for yeast work, where would you recommend going? Apart from, joining your lab.
Yeah.
Not all of us are cool enough to live in Vancouver, Scott.
So one of the things that I envisage once the DNA is printed, I would like to actually build a community. I've got ideas of like a website where it's like a repository, a bit like the Open Bioeconomy Labs, or we might, if Jenny's willing, be able to post our parts up there, because I can see this as being very important for people, and learning would be, it's really important. So there's also, Jenny talked about reclone.org, so there'll be a lot of stuff mainly about OpenEnzyme, but I'm certain that you'll be able to get a lot of questions answered there about other, about the yeast. And, there are people that are producing enzymes in yeast. There are some enzymes you can't produce in E. coli. So this kit would help with that.
I think we're at time. I'll hand back over to Eduardo. Thank you.
Okay, thanks everyone.
Thank you, Scott, so much for being here and sharing this with us. I see people are active on the notes, so maybe that's a good way to keep connected. And I know there are people very interested and probably with more questions for you. So definitely, or there is lack also. So please stay in touch. Thank you so much.
Thanks everyone.